So a hugely warm welcome to everybody for joining us this afternoon. We're really excited to welcome Professor Simon Carding back. We met Professor Carding about, I think it was 3.5-ish years ago. And so we're delighted to hear about how his research has been progressing and what he's got to share with us today. Just a few little bits of housekeeping. This session is being recorded and we will be putting it out onto, I want to do something else. I'm just waiting 1 second for it to go live. Eliane, could you help me out? It says I do not have permission to live stream it.
Wait, are you logged in with your normal Facebook account on Facebook?
I'm logged in with my account to the page. So should I be logged in?
Yeah, no, normally that's fine. So where are you at? What steps have you done already?
So on the Zoom, I've clicked on live stream to Facebook, and then it's asked me which page, and I've chosen that.
Yeah.
Select page, and it says go live, and I go live, and it won't let me. And in the good old days, it used to work.
Yes, that's strange. We tried it out and didn't think that last step would be the problem. I don't know, because you're the host, normally only the host can do that. So
okay. My thought is to keep going with the meeting and then I'll try and sort it. Is that okay?
Yeah, maybe try again later.
I'll try again. Okay. Right. Let me just start that again. So welcome everybody to this afternoon's session, part of our Science to the Sofa series, and we're very, very delighted to have Professor Simon Carding with us this afternoon, who we met 3.5 years ago, and we're really looking forward to seeing what progress has been made on his research and what things he has to share with us today. This session is being recorded and we hope it'll be live streamed on the Facebook page. Just to reassure everybody, we won't be able to see anything of your screens or your names either on the recording or on the Facebook Live page. If you want to be entirely anonymous for this Zoom chat, then please change your name by using the rename button if you've got any reason to not want your name to be known by the other people in the Zoom room. So it would be great to know where everybody's from. I know when we put this out on social media, we had a lot of very, very interested people from all sorts of places. So please add something into the chat which says hi and where you're from, because that's always lovely to know. And I'm just going to introduce both myself and then move on and introduce Professor Simon Carding. We're hoping this session is about 45-ish minutes, 45-50 minutes for a talk, and then we're going to be running a question and answer session. So please feel free to post questions in the chat, which we'll pick up and ask on your behalf at the end of the talk. And please feel free to post questions on the Facebook page from us. So my name is Carolyn Leary. I'm co-chair of the Sheffield ME and Fibromyalgia group, which is a voluntary group run by patients and carers in the city of Sheffield and covering South Yorkshire and North Derbyshire. This talk is being jointly organised with Cinnamon Days, CIC, Community Interest Company, and delighted that both Isabel Hemmings and Sue Wharton are here. They're two nutritional therapists who are co-directors of this social enterprise, which runs the Nutrition Club for ME, CFS and long COVID. And we'll hear a little bit about that at the end of today's session. But please put in the chat if you put your email in the chat if you'd like to be emailed with any information about that Nutrition Club. So I am absolutely delighted to give a very warm welcome to Professor Simon Carding. And Professor Carding is leader of the Food Microbiome and Health Programme at the Quadram Institute, which is in Norwich, and is also Professor of Immunology at the University of East Anglia. And if you look up his bio, it's significantly longer than that because he has a huge number of achievements to his name. And well, how exciting! Somebody is here from Trinidad and Tobago. Yeah, just mention that. So, can I hand over, please, to Professor Carding?
Sure. I will share my screen. Can you see the
We
screen?
can see.
Can you hear me? Yeah. Okay.
Lovely.
Well, first of all, thank you, Carol and Isabel, for the invitation. It's nice to be back. I wish I was in Sheffield. It's one of my favourite cities, but second best is virtually. I'm virtually there. So what I'm going to talk about in the next 45 minutes or so is give you an overview of some of the things we're doing at the Quadram Institute to advance research on ME/CFS. So for those of you not familiar with Norwich and the Norwich Research Park, I've put up this picture here which shows Quadram as one of 7 institutions in the Norwich Research Park. So here we are. We're next to the Earlham Institute and the John Innes Centre, which some of you may know for its research on plant science. We also have the University of East Anglia and the Sainsbury Laboratory. And we have Norfolk and Norwich University Hospital, which is one of the biggest and busiest hospitals in the UK. And within the Quadram itself, there we go. This is the sort of schematic of the Quadram Institute. We've been in the building for about 3 years now. So you can see that it's a split between the NHS and research, the Quadram Institute. So in the basement here, we have the endoscopy unit of the hospital, which is one of the busiest in Europe. It sees about 40,000 patients a year, employs a large number of people. And above that, we've got a clinical research facility, which allows us to undertake human studies. And then we have 2 floors of laboratories, including a high containment laboratory for working with really nasty microbes. And there, so in total, the building costs probably around £80 million now, I think. And that can house, we've got about 250 scientists in the building. Keep pressing the wrong button here. Sorry about that. So this is the Institute's vision. It's to understand how food microbes interact to promote health and prevent disease. So not surprising, my own interest in gut microbiology, immunology is sort of central to that vision. So we're all focused on either food, where in the gut it breaks down to release the nutrients, how the microbes interact with our food, and how that then leads to health, or in some cases, disease. So what I'm going to cover in my talk today is basically uncovering disease mechanisms that will allow us to identify novel ways of treating ME/CFS. I'll talk briefly about some of the fundamental research which we are doing, which is focused on viruses in ME, autoimmunity, immune senescence, which is a new area of interest to us and may actually link to infections and viruses. And then I'll talk about translational work, 2 clinical trials that we have planned, one that will start early in the new year and one that will start in the middle of the new year. I'll talk about both of those. And then I'll talk about how we're trying to develop collaborations beyond the Norwich Research Park to involve our European collaborators and colleagues. And this is the European ME Research Group, and a new initiative is the Young ME Research Group, which is to promote research among younger individuals to make sure that we've got people in the future that are interested in working on ME. And then I'll talk about some of the things we're doing to try and influence the people that control a lot of the money in the UK and Europe as well. So how we're influencing government and funders. So just sort of context, I think ME/CFS now dates back about 90 years. It's been associated with various sort of so-called epidemics. The first one being in 1934, when it was misdiagnosed as polio. Since then, there have been other outbreaks occurring in Iceland, in London, and elsewhere in the US. And by 1990, we had over 60 outbreaks. And during this time, how we describe ME and how we distinguish it from other diseases has become a little bit clearer. And the Center for Disease Controls has identified it now as being an important priority 1 disease that needs to be addressed. And along with that sort of an idea of it being an infectious disease, I think most patients will report a pre-illness onset that's maybe attributable to their symptoms and the persistence of their symptoms. And some of the viruses that are sort of the usual suspects, if you like, are shown here. So you'll be familiar with these, I'm sure. Epstein-Barr virus, herpes viruses, parvovirus, enterovirus, and cytomegaloviruses. And maybe now even SARS-CoV-2 is linked to onset of ME. ME is a complex disease, and this makes it difficult to sort of untangle the causes and the pathways that are responsible for developing the disease. This slide just illustrates a lot of potentially interacting factors that contribute to the development of ME, including immune dysregulation, energy issues, mitochondria impairment, intestinal dysbiosis, changing gut microbes, impacts in the central nervous system, and then SARS-CoV-2 infection and long COVID, which may lead to symptoms that resemble ME/CFS. And I think a current question that's yet to be addressed is long COVID and ME/CFS, are they the same, or does one lead to the other? And on this slide, I've identified or tried to summarize overlapping symptoms and some symptoms that distinguish ME from long COVID. And I think one thing that's driving a lot of research into long COVID is the significant number of people worldwide that are affected by long COVID, in excess of 65 million people. And the economic impact of this in America is considerable, $3.7 trillion over the next 5 years, economic cost. And interestingly, there's around anywhere between 13 and 45% of long COVID patients that will have a diagnostic criteria that meets that of ME/CFS. The impact of this potentially in the UK is that the number of sufferers from ME/CFS could increase to more than 1 million if these long COVID patients are included in diagnosis. So this will be a huge impact on the disease burden in the UK of ME/CFS. An important point at the bottom here is that a lot of these features or symptoms are also present in other infectious illnesses and other non-infectious illnesses, such as post-critical illness syndrome or post-intensive care unit syndrome, and including heat stroke. So a lot of the symptoms we're describing here could be the consequence of an ongoing or a previous infection, be it a virus or some other microbe. So these seem to be very overlapping, but there are, say, some distinctive features. The painful lymph nodes, chemical sensitivities, tinnitus, which are not present in all ME/CFS patients, but they're usually absent in long COVID. And I think the majority of ME/CFS patients don't suffer from rash or hair loss or decreased smell and taste. So very similar, but probably distinctive disorders. So the question we've been asking is the origin of ME/CFS in the gut, and are there microbes in the gut which can trigger or cause ME/CFS? So just a couple of slides to introduce you to gut microbes. So when we say the gut, we mean the entire gastrointestinal tract from the mouth to the rectum is included in that description. Within the gastrointestinal tract, there are trillions of microbes made up of not just bacteria, but also viruses and non-pathogenic fungi. And these play key roles in keeping us healthy. By far, the largest population of microbes of our human microbiome is found in the large intestine. And that makes up of 95% of all the microbes found on or inside our body. So that includes on the skin, reproductive tract, internal surfaces. Vast majority reside in the large intestine. And the picture on the right here shows you some of the microbes that can be identified in the gut, usually in stool samples. This one here is SARS-CoV-2, which has been isolated from a human stool. sample. So a very complex ecosystem that functions with us to keep us healthy. And some of the key things that the gut microbes are responsible for are digestion, food processing. They are a source of essential nutrients that we cannot synthesize, including vitamins. They play an important role in defense. They act as a physical barrier to stop infectious microbes gaining a foothold in the gut. And they also stimulate the immune defenses. In particular, antibodies are secreted in the gut to bind to and neutralize pathogens. And then they also play an important role in actually the development of the immune system itself and of the brain. So animals, and these are usually rodents that are born in a sterile environment and never get colonized with microbes, they have significant deficiencies in their immune system and in the development of their brain. Also, we now know there's the gut-brain axis, and microbes in the gut can influence the brain in turn that impacts on our behavior. They can regulate appetite, for example, food preferences. They can influence mood and depression and therefore mental health. So gut microbes are increasingly being considered as a key element to keeping not just the gut healthy, but the rest of us, in particular the brain, healthy. The other side of this is that changes in the populations or the function of our gut microbes are increasingly being linked to a whole array of diseases. And in fact, some people would argue every disease that we know of is associated with changes in our gut micropopulations. And this slide just illustrates some of them. So if we come heart disease, atherosclerosis, thrombosis, skin, psoriasis, skin cancer, metabolism, obesity, type 2 diabetes, cancer, lung, in terms of lung cancer, obstructive pulmonary diseases, asthma, and then the gut-brain axis as well, where there's links to the development of neurodegenerative disorders, the dementias, and psychiatric disorders as well. So it's becoming increasingly apparent that we need to be studying the gut microbes in patients with disease to see if there's alterations, if these might be actually part of the development process of some of these diseases. So the question is, what has this got to do with ME? So is there a gut origin for ME/CFS? There are several lines of evidence which suggest this is correct. So most patients will have significant GI symptoms preceding or coincident with disease. There's been reports of high prevalence of virus infections in patients that originate in the gut. Patients have a leaky gut wall, which allows microbes and microbial products to get into the circulatory system, to spread through the body, cause inflammation, tissue injury. This is accompanied by increased sensitivity for food. And we now know that there's a consistent evidence of changes in our gut micropopulations that are reflected in decreased diversity. The numbers and types of bacteria is decreased. There's an increase in harmful bacteria, decrease in beneficial bacteria. So the gut microbiome is a useful target, potentially a useful target to address some of the issues related to either cause or severity of ME. And what we're particularly interested in is asking the question, is ME a consequence of chronic microbial stimulation of our immune system, which causes our immune system to prematurely age or become exhausted, which then can lead to a lot of, or explain a lot of the symptoms that ME patients suffer from. So there's a lot of interest in manipulating or changing our gut micropopulations for not just maintaining health, but for treating diseases. And at the Quadram, we are interested in using a procedure called fecal microbiota transplantation, or FMT, as a potential treatment for ME/CFS. Now, FMT is not new. It's been part of Chinese medicine for many, many centuries, dating back probably to the 4th century, when it was used to treat diarrhea and food poisoning. And then by the 16th century, there was a concoction called yellow dragon soup, which was used to treat a variety of symptoms, including fever, vomiting, constipation. Veterinarians have been carrying out FMT since the 17th century, and this picture here shows an ill animal being treated by administering of a stool sample from a healthy animal that's delivered directly into the lumen of one of the stomachs of the cow. During the Second World War, German soldiers were administered what was called camel poop soup, which is an effective treatment for dysentery. And there is now this product which can still be bought, which is the active component of the camel poop soup called Bactis subtil, which is available particularly in Europe as a preventative treatment for all sorts of GI ailments. The first real use in clinical trials was in 1958, when it was used to help patients recover from surgery. And a little later, it was used to treat patients with enterocolitis, 1958. 1989, it was being used to treat ulcerative colitis and other inflammatory bowel disorders in Australia. And then the real breakthrough in terms of changing clinical practice in the NHS occurred after 2013, and a report that a very difficult infection, recurrent Clostridium difficile or C. difficile infection, could be cured by FMT. And the cure rate was in excess of 90%. And this has led to it being now widely adopted in the NHS as a frontline treatment for recurrent or difficult to treat C. difficile infections. So this is changing clinical practice, and this is likely to be the forebearer of other uses of FMT in the NHS. So as FMT's evolved, it's moved through different routes of administration. Originally, it was delivered via an enema, then various tubes, and now we are using capsule delivery. So these will be capsules of a similar type, shape as night nurse or day nurse capsules, for example. And in terms of the future, this could be applicable to a whole array of disorders and diseases which are associated with changing gut micropopulations. And this could become very personalized. So the formulation of the capsules could be designed to meet the needs of a particular patient and to replace the lost beneficial microbes in that individual. So we've been conducting FMT at the Quadram Institute for a few years now, been working with the Norfolk and Norwich University Hospital to treat patients with recalcitrant C. difficile infection. These are a little bit dated. We've now done nearly 40 patients, and the success rate is in excess of 90%. The failures are attributable to other unrelated illnesses in patients. So antibiotic administration was one of them. And what we're currently doing is constructing a super clean facility to comply with recent regulations imposed by the MHRA so that we can use this more widely as a treatment. And in the future, we'll be running trials in ME/CFS that I'll talk about in a minute. And originally, this was the route of administration. We prepare this fecal slurry donor in a syringe, and then this would be injected into a tube that is inserted into either the rectum, the colon, or the small intestine. Now, we're using what we call capsules to deliver this. And this is equally as effective as delivering it through a tube. So it's a much less invasive procedure. Evidence that FMT can work in ME/CFS patients comes from pioneering studies carried out by Thomas Barodi in Australia. He's treated 60 mild to severe patients, 52 of them. Most of them had irritable bowel syndrome. And this was transcolonic infusion plus rectal infusion. Of the patients treated, 42 responded, reporting improved sleep patterns, less fatigue, less lethargy. Interestingly, a majority of them were also symptom-free 15 to 20 years after follow-up. And most, if not all of them, had resolution of their GI symptoms. So these were individual patients, and these need to be substantiated in a clinical trial, which then leads us on to the clinical trial we're proposing to do at the Quadram Institute, which is called Restore Me. It's a phase 2 clinical trial designed to test or evaluate whether FMT will restore a healthy microbiome in patients and improve their physical and mental health. This is the study team and design. It involves a lot of different people with a lot of different specialties that are coming together to help this trial. So we have primary care specialists, the consultant microbiologist, environmental science specialist, gastroenterologist, etc., aging and dementia research, A lot of people involved in this. It will be a single site, randomized, double-blinded, placebo-controlled study. Capsules, which we've called Neptune capsules, will be used to deliver the donor samples, and we're testing efficacy and safety. This trial is going to be distinctive from most other clinical trials carried out in ME, CFS, because we're using objective outcome measures. And rather than questionnaire-based, patient-completed questionnaires, we'll be measuring physical activity using accelerometers. We'll be assessing sleep objectively with help of the UEA Sleep Center. Physical capacity will be assessed using a nuke-based objective and questionnaire, quantitative questionnaire, we call it FUNCAP. And we'll be looking at brain imaging and vision and cognitive function using clinically validated objective measurements as well. So in addition, this will be the first clinical trial to test FMT in ME patients. So it's an important trial on several fronts. This is a cartoon of the design. So we're planning on recruiting around 120 patients via East Coast Community Healthcare, there's Chronic Fatigue Syndrome Center, excuse me. The patients will be divided into two groups, placebo and treatment. Treatment will be through capsules, then we'll monitor them at 2 time points, 3 months and 6 months, assessing the accelerometer, cognitive tests to assess the impact on cognitive function, physical activity, and then we'll be taking fecal samples, blood samples, nasal swabs for looking at various aspects in research projects associated with this. So this is the timeline. We were hoping the trial would have been running now, but we suffered more than a 2-year delay because of COVID. And also the MHRA decided that human stool was a medicinal product, which means it had to be produced in accordance to the principles of the GMP, which is super clean facility. So we've had to design and build a super clean facility for handling and processing stool to manufacture the capsules. So we're currently we've completed the construction of the facility. It's now going through its stress testing. We'll wait on an MHRA inspection to say, yes, you can use the facility. We'll give you a license, which we're hoping will be early next year. Then we can apply for the trial license and get ethical approval, all with the aim of trying to get recruitment underway to allow us to start the trial in June next year. So that is the current timeline. Running the trial gives us an opportunity to utilize the samples we're collecting to undertake new research into the causes of ME CFS. And one of these is being carried out by a PhD student who's about to graduate from my group, Rick Hargmans, who's interested in human gut viruses in ME CFS. And so the virus is particularly interesting, the herpes virus and the enteroviruses that have a long association with ME CFS. So these are very common. They can reside in a dormant state in most of us and become reactivated in response to various stresses, be an infection, changing diet, medications, etc. They're very good at hiding in the body, hiding away from the immune system, so they can reside in the body for decades without being detected. But they do become reactivated under stress, a variety of stresses. And they have a history of being associated with the gut. So it could be reactivation or spreading of the virus to the gut that could be part of the developing symptoms in ME. So while we wait for the Restore Me study, Rick has been working with a Norwegian group in Harstad, Norway, who have undertaken their own FMT study in ME patients called the Comeback study. This will be completed at the end of the year. This involves 80 ME patients, placebo and treated, 40 in each group. They are administered the FMT or placebo via an enema, retention enema, and then they are assessed at 3 months and 12 months. This is using a fatigue scale, so questionnaires, patient-based questionnaires, and research samples have been collected, and Rick is working with these samples to look at what impact FMT has on some of these viruses with a known association with ME. So this was part of his ME project. So looking at the gut virome using high-powered sequencing analysis. And he's been working with Dr. Martin Carr and Adam Brooks to look also for the presence of other gut pathogens in these individuals, pre and post intervention. So this feeds nicely into Restore Me, in that we can learn a lot from the comeback study that will help us improve the design of the Restore Me study. And we've received funding from ME Research UK to allow Rick to extend his gut virus studies into the Restore Me study, where so he'll be looking at gut virome analysis again by the sequencing, looking at also at viruses in the blood, and working with one of our collaborators at the University of Valencia, we'll also be looking at viruses that are found in tissues rather than the blood, as well, to see how they respond to FMT, or if they're activated or inactivated in ME. So that's a separate, if you like, spin-off project, which is feeding into the Restore Me project. One of the other projects is led by Dr. Catherine Seaton, who's an Invest in ME Research Fellow. And she's interested in gut microbes, the impact this has on stress and the immune system in ME. And so center of her project is looking at accelerated aging in the immune system in ME CFS patients. She's received some money from Solve ME, the big American ME charity. And this is the question that she's asking. So as we age naturally, our immune system loses functionality through various reasons, causes. And this means that as we age, we become more susceptible to infections, we respond less well to vaccinations, and we can develop chronic inflammation, autoimmune disorders as well. So ME/CFS, is this an accelerated process? That's one of the questions we're asking. And can FMT actually reverse some of these aspects of aging of the immune system? So can an FMT reverse premature immune aging and restore immune function in ME patients? So these are the aims: confirm immune aging in ME patients and link that to symptom severity, and then determine whether FMT impacts on immune function and disease severity in the ME patients. The experimental approach is a little bit complicated, but it means essentially taking blood samples and looking at the different populations of immune cells in the blood, looking at their populations. Are there changing populations? And are there, more importantly, change in function, particularly the immune cells are responsible for eradicating viruses. So natural killer cells are of particular interest because of their ability to kill virus-infected cells. And again, there's been reports of defective natural killer cells in ME CFS patients. So the impact of this particular study is that we'll understand more clearly the link between our gut microbes and the immune system in ME. One of the other questions that Catherine will be addressing is the microbiome, and is this a driver of oxidative stress? So oxidative stress is the result of change in the balance between the production of materials, compounds, metabolites that act as antioxidants versus those that cause oxidative stress, which we call free radicals. Free radicals and oxidative stress can lead to tissue injury and inflammation. So maintaining this balance between antioxidants and free radicals is very important. The source of natural oxidants to us come from our diet, principally, and our microbiome, which processes our diet. Free radicals are derived from, not surprisingly, environmental sources, pollution, stress, smoking, ultraviolet exposure, etc. So it's the balance of these that's important to maintain. So the microbiome is potentially a source of antioxidants as well as free radicals. So in the inflamed gut, for example, the balance would be tipped to free radicals. However, if the microbiome is able to process foods to produce important antioxidants such as vitamins, minerals, short-chain fatty acids, then this tips the balance towards the antioxidants and less oxidative stress. So evidence that this is changed in ME patients. So ME triggers and can include GI infections which produce free radicals. ME CFS patients have irritable bowel syndrome, which again results in the release of free radicals. And the leaky gut, of course, will allow toxic material in the gut to get into the bloodstream. And some of these, of course, will be free radicals, which can cause systemic problems throughout the body. And we now know from analyzing the gut microbiome in ME patients that they have reduced levels of antioxidants. In particular, short-chain fatty acid producing bacteria are deficient in ME CFS patients. So I think this provides some evidence for the importance of maintaining the balance here between antioxidants, free radicals in ME. So this will be part of Catherine's project. And what she'll be doing is confirming the gut microbiome is actually a source of oxidative stress in ME patients, and looking at whether or not FMT can remove the source of free radicals in the gut to alleviate some of the symptoms and certainly oxidative stress in the patients. So that's all related to the FMT, the Restore Me trial. One other trial that will be happening a little sooner is called a red light therapy or photo biomodulation trial. And this is based on evidence from other conditions where low-level light therapy, or PBM, is a safe and effective treatment for pain, inflammation, edema. It's approved by the USA Food Drug Administration for the treatment of acne, muscle and joint pain, arthritis, blood, and circulation issues, and hair loss. So this could be a potential application for chronic fatigue syndrome and ME. So the question is red light therapy a promising treatment for ME CFS? And is there a cascade of beneficial cell responses which can be achieved with low-level light therapy? The way it works is that it acts on mitochondria, which are the energy-producing units within all our cells. And so the red light, if you like, kickstarts or reboots mitochondria to produce more energy. And in this regard, it's a chemical called ATP, which then allows organs and cells and tissues to work more effectively. And we know that ME patients have mitochondrial abnormalities, and this compromises their ability to produce the energy substrates that lots of our cells, tissues, and organs need. So we were planning on exposing ME patients to red light with the aim of then re-energizing our mitochondria, so the end mitochondrial batteries go from being empty or half empty to full. And this then will have some positive benefits in terms of activity, cognitive function in ME patients. So the aim initially, this will start with a small scale study to evaluate the acceptance, adherence, and safety of PBM therapy. This has not been trialed properly in ME patients before, so we need to ensure that patients are happy to use the lamps, that it's safe for them. And also we're developing measures of the efficacy or treatment using questionnaires, cognitive assessments, and risk-worn activity monitors, all of which will feed into the Restore Me trial in terms of how well these measurements are accepted by patients. So this is study design. I said initially it's a small number of patients, 10 patients initially, to evaluate this. It'll be a remote study, which is an important consideration, particularly in terms of reaching severely affected patients that are house and or bedbound. Online assessments, posting equipment to participants. And it's that way it's designed to be more inclusive for ME patients. They are not required to come into the institute, visit a GP surgery or anything. This all can be done remotely. And the impact, if this is a successful, we will be extending this into a much larger clinical trial with many more patients. And as I said, a lot of the measurements and assessments will feed into the use of the assessments for the Restore Me trial. Again, it's a multi-purpose team, so we'll be working with one of our collaborators, Christian Sommerfeld, University of Bergen, Glenn Jeffrey from who's a vision expert, University College London, Michael Hornberger, dementia expert, and Andrew Atkins here at the UEA, who's an exercise expert. So I said it's 10 patients initially. Eligibility is quite simple. We've obtained ethical approval, so we're going to start recruiting next month. So this will happen quite soon. As I said, it's a remote study. We're using an online tool called Mantle, which will allow us to collect, administer a lot of information about how the patients are responding to this, in terms of their mental and physical health. And as I said, no study visits. We will post monitors, lights, etc. to patients. So that's a bit of a whirlwind of what the type of research, both translation and fundamental research, we carry out at the Quadram. I did say I would mention something about how we're trying to get more money for ME research, not just for us, but for other people that are trying to do ME research. So we are part of a research working group that's been established by the Department of Health and Social Care in the UK that's been tasked with developing an ME CFS delivery plan. How, where can we invest, or the government invest, to improve research, but also patient care for ME CFS patients. And I'm also part of an American equivalent of this, which has been carried out by the US National Institutes of Health, which is coming up with its own ME research roadmap. And I'm a member of the working group there for chronic infectious disease and the immune aspects. And we're trying to get in the face and in the ear of MPs to try and influence them and make them aware of what ME is, how bad it is for patients with ME, and how we desperately need more money to put into ME research. So these are just some of the people that we've been talking to. We have more MPs visiting throughout the year. So we're really trying to impress on MPs the importance of supporting anything that comes out of the DHSC recommendations, which will be, I think, first quarter of next year. And then we're trying to work with ME researchers outside of the UK, particularly in Europe. So I'm a member of the European ME Research Group, Emerge. The flags here just indicate where the members come from. There are members coming from other countries as well, which I don't have on here. And I co-chair this with an individual from Denmark. And what Emerge does is it participates in trying to influence policymakers by being representation on important government bodies, funding bodies. We're coming together to develop an EU Horizon 2020 proposal. We've just submitted a stage 1 application that involves 10 centres across Europe. I represent the UK in this, and we're expecting to hear back before Christmas on this. If we're successful, we'll advance to the next stage, where we'll be competing for around 8 or 10 million euros to undertake both fundamental and clinical research in ME. We're also providing advice for developing new tools for improving the diagnosis and outcome measures. Clinical colleagues on Emerge are involved in that. And we're also collectively advising on clinical studies and trials, including Comeback and Restore Me. And every year we organize an ME colloquium where we bring researchers from across the globe, basically, to the UK to talk about their latest research, to try and again promote collaboration rather than competition among ME researchers. Young Emerge. So a lot of us are not getting any younger, so we need to make sure we've got young, bright young things that are keen to work in ME, to sustain their careers as ME researchers. So that is the central premise of Young Emerge, to encourage research in ME by establishing networks of early career researchers. These are PhD students, postdoctoral fellows, contribute to new research, publish work on this, organize annual meetings on this. So these are the key people. So 2 people from my group are involved in this. But there's also an individual from Norway and from Spain that are organizing, coordinating activity. And 2 of these people have been invited to a meeting in Washington, DC, where, not surprised, the Americans are copying us, and they're setting up their own version of Young Emerge. And they want to learn from Catherine and Rick about how they can do this most effectively. Finally, if you're interested in contributing to our research, I would encourage you to look at this Quadram website. We have a volunteer database. We encourage you to sign up for that, because as more studies come online, we will need to recruit participants. If you're on a database, that greatly facilitates recruitment of patients. And my very last slide, so this is to sort of offset questions I often get, is how can we maintain a healthy gut and mind? And what I put up here is my car analogy. So if we consider food is the fuel, gut and our microbes are the engine, and the brain is the transmission, and then the body functions are drive shafts, wheels, and exhaust is the waste. So how can we get this to work effectively and have a super running car? Well, the food quality is important here. The more fruit and vegetables you eat, the better. The more color you have on your plate, the better. Less processed food. Adopt and sustain a healthy lifestyle. Promote your health. And if you do that, your body will function like a Jaguar sports car. If you don't, then you could end up running around in Del Boy's Robin Reliant. So it's a crude analogy, but I think it works, and it shows you, hopefully how you can have your body purring along like a Jaguar and not rattling along like a Robin Reliant. And with that, I will thank you for your attention. I'm happy to answer as many questions as I can. And just to remind you that all the research we've done is provided by funds like yourselves that make donations to charities, which in turn support our research. There's very little government money here. It's all coming from charitable donations. Thank you very much, and I will stop sharing.
Excellent. A huge thank you to you, Professor Carding, for such an incredibly detailed and comprehensive and just really, really clear overview of where you've got to. And certainly I'm spotting an awful lot of things that have really moved on and changed since we last spoke to you. So a massive thank you, I think, on behalf of absolutely everybody in the wider ME community for your dedication to this area of work, because people like you are in very short supply, as you know. So
that's unfortunately true. Yes,
it's so appreciated. Right, I've got well, what I can only describe as a shed load of questions. So
okay.
I'm hoping that with the help of Isabel and Sue, my companions in this talk, that we'll manage to cover almost everything that people have raised. I was going to start off with a sort of timeline question from Lisa, which is, given how long it takes to reach a diagnosis of ME/CFS, how can we know if the gut issues caused or were caused by the ME/CFS, given that kind of diet is one of the things that can go to pot a bit when you first get ill and you're really tired and you don't eat properly and things. So there's a kind of, chickens and eggs question, really.
Yeah, so that's a very difficult question to ask, because in an ideal world, if you had enough people, you would want to administer the infection and then follow them through time. Of course, that's unethical. So what we rely on is patient histories and patient records. And, it's whenever I talk to ME patients, I'm just amazed by how many say, oh, I had EBV, or I had herpes, or I had a virus infection, flu even. And after that, I just never seem to recover. And then that led or progressed to ME/CFS. Now, one of the best clues we've got actually comes from SARS-CoV-2, because we know the trigger there is the virus. These people were presumably healthy before they acquired the virus infection. So following people that were infected with SARS-CoV-2 and seeing how many of those develop ME and how many don't, you've got probably what is the best natural experiment there to relate symptomology of ME back to that virus infection. Now, the thing to bear in mind is that getting infected by one virus makes you immune compromised and vulnerable to other virus infections. So again, it may be multiple infections which ultimately lead to ME/CFS. And I think the longer it takes to recover from a virus infection, the more susceptible you come to other infections or insults, which could then accumulate into long COVID ME/CFS. So it's likely, and it's the best potential trigger we have currently, is a virus infection. And all the viruses we've been talking about all can live in the gut.
Interesting. Yeah, I think we really, really don't understand that fully do we all really acknowledge that fully? One of the, you've obviously talked about viruses that compromise people's immune systems, but we've had a number of questions about how medications compromise people's gut biome and therefore their immunity. So obviously antibiotics is often talked about, so you might say a little bit more about that, but are there other medications as well that potentially compromise?
Absolutely. So the important point to remember is about 80% of all medications are oral. Now, your gut microbiome will treat anything you consume as a potential food source. So drug, food, it will try and utilize that or change it in some way so it can be accessible to them. So antibiotics, yes, are a good example of how you can devastate your gut microbiome. And, you know, depending on how many and for how long, it can take months or years to fully recover. But there are lots of other drugs like painkillers, anti-cancer medications, which as they get processed by your gut microbiome can make them essentially ineffective. So they become useless or much less effective, or they can actually make toxic products which are harmful to you. So the various ways your gut microbiome, depending on what your gut microbiome is, it could make a drug useless or it could make it even more dangerous by making toxic products. Now, there are good examples of beneficial effects of your microbiome processing drugs. Metronidazole is a good one. That's we take as a prodrug, inactive. It requires processing by our gut microbes to become active. So there are lots of facets to this. And basically, your gut microbiome, your own gut microbiome, which is unique to you, will be the determining factor in how well the medications work.
Okay, and following on from that, this is also from Deborah, thank you, is if we think our systems have been compromised in those ways, is there anything that we can do, thinking about your car analogy, to kind of turbocharge the recovery?
Yeah, so obviously having a healthy diet is good, but I would say fermented foods are superfoods because you get the benefit of the plant material, but you've also got the benefit of probiotic bacteria, which are part of the fermented food. So things like sauerkraut, kefir are good because you're getting double benefit from them. So probiotics, you can go into supermarkets and buy Actimel, Yakult. I would say money better spent on fruit, vegetables, fermented foods.
Interesting and extremely clear. So thank you. We've certainly had questions about what are recommended supplements. So I think you've probably just summarized your view on those. So that's great. Can the gut microbiome be affected when people have things like gut dysmotility, like gastroparesis and other?
Yes. So if you look at the two extremes, sort of diarrhea, constipation, and everything in between. So diarrhea is usually a response to try and eliminate something which is noxious, toxic to the body. Best example of food poisoning. The body tries to get rid of that contaminating pathogen in your food as quick as it can. So you get diarrhea. Constipation, on the other hand, can be caused by various things, physical, anatomical issues, dietary issues. But the problem with constipation is that toxins that would normally be expelled from the body build up because there's less transit. And some of those, particularly in ME patients who may have a leaky gut, now can get into the gut. So ideally, you need to be somewhere in between because you can have issues at both extremes. And certainly, if you have any physical abnormalities in your gut wall that affect innervation or motility, then that could have knock-on bad consequences for your gut microbes and in turn your health.
Interesting. And following on from that, I haven't noted down who asked it, but is there any connection between a weak gut, whatever that means, and insomnia? Can it affect a whole range of other things like sleeping?
Yes. So, I mean, I talk about the gut-brain axis. Now it's becoming increasingly clear that microbes in the gut can influence the brain in various ways. So signaling in the brain, which impacts on behavior. So I talked about how it can influence appetite control. It can also influence the production of neurotransmitters in the brain, which will impact on an array of functions. In the extreme case, that could be linked to the dementias, autism, for example. Milder effects could be depression, changing beneficial behavior like sleep, everything. So yes, there are a variety of ways this can happen. We haven't identified how, but we're getting some significant clues, and they are probably related to metabolites, so the products of the things that microbes produce. So if they get into the bloodstream and they get into the brain, they can change structure of the brain and the way the brain works. And that, again, has lots of consequences for behavior.
It's just extraordinary, isn't it? How incredibly powerful it is. We've also had a couple of, somebody's asked about, could problems in the gut affect just one side of your body only? And is there any link between a poor or weak gut and fascia tissues?
Not aware of any,
Okay.
because if you think about how the products that gut microbes produce get around the body, you know, it's through the circulatory system, through the lymphatic system, or actually directly through the nerves that connect the gut to the brain. So I think the only scenario I could think of is if you had a shutdown or restrictions in some blood vessels would affect certain parts of your body, or that might have an impact there, yes. But I mean, they have free roam around the body. If they can access the circulatory system, lymphatic system, that gives them access to virtually every part of the body.
I mean, certainly, yeah. I mean, from our limited knowledge, anybody with difficulties down one side of the body should actually seek a medical.
Absolutely, absolutely.
Probably quite urgently. So yeah. We've got a question about is there a microbial difference between taking Prapsule, love the name, and a pro or prebiotic? There's lots of people take, well, this questioner says lots of people take pre and probiotics, but find they have some limited effects.
Yes, so I think I told you about my bias here.
Yeah.
You know, spend your money on prebiotics, not probiotics. You know, on the food, think of feeding your healthy gut microbes. And that is basically the government's guidance, 5 a day, 5 servings of fruit and vegetables. That is the food that your beneficial gut microbes need to keep healthy. The issue with probiotics is that when you go out and buy Yakult or Danone, they contain one, maybe more, 2 or 3 organisms, but each product contains different bacteria. And so it's very difficult to compare one to the other. Question you need to ask is, well, if I drink this yogurt pot, how many live bacteria are in there and how many are going to survive, transit through all the stomach acid, and everything else to actually get to where they need to be? So of course, from a commercial point of view, well, they'll just say, well, you need to take it every day for ever. And that way it builds up. Well, yes, that makes a lot of money for certain companies, but with a capsule or with fermented foods, you're getting a whole cocktail of microbes. And, you know, you think about the complexity of all your gut microbes, it's hard to say, well, one microbe is more important than another. They all work together and talk to each other to collectively keep us healthy. So in that regard, it sort of makes sense. Well, you're going to need more than one to make any change. You're going to need, you know, a consortia, a cocktail. So the FMT is an extreme case of that, where you get rid of your bad microbes all in one go and you replace it with a healthy one. But over time, consuming prebiotics and fermented foods, you're going to get that benefit as well, but not from single-use probiotics. Interestingly, probiotics are used clinically to treat, for example, like bladder cancer, but that is a super cocktail of 7, 8, 10 different bacteria only available by prescription. Single probiotics unlikely to work.
Okay. So perhaps that also addresses Sylvia's question, which is that when you're doing the prep for an endoscopy or similar, how do you repair things afterwards? So it sounds like mass consumption of prebiotics.
Yes, that was certainly up. So with FMT, the pre-procedure is bowel cleansing. We used to give antibiotics, but that was unnecessary. So now it's just a bowel cleansing prep, which you would get for endoscopy, colonoscopy. You'd be asked to take a bowel cleansing. And that's based to make space for the new microbes to allow them to establish much faster. Yeah.
Okay. We've got a whole set of questions which relate to your research. For example, if you have severe allergies to many food, would you anticipate any sort of problematic reactions to FMT?
That's interesting. That is very interesting. Yes. So I would think if your gut microbes are responsible for directing your immune system, helping to make the immune system work more effectively and to mount responses to things that could potentially cause harm, but not to those which are not harmful, like food, for example. So if you eliminate the gut microbes that are presumably driving immune response to react to the foods, an FMT might get rid of them. And that may, this is theoretical, resolve food allergies and maybe some other disease as well. So interesting studies in obesity. Okay, so you can transfer the microbiome from an obese human into a mouse and it will become obese. But if you transfer the microbiome from a lean human into a mouse, it stays lean. If you co-house those two mice, the lean mouse protects the other mouse that got the obese microbiome from becoming obese. So we might expect other unforeseen benefits of an FMT in terms of influencing metabolism, preventing metabolic disorders like obesity. These is all theoretical, but interesting questions which we can ask in FMT trials like the Restore Me. And there may be unforeseen benefits we just haven't thought about yet. But yeah, the food allergy is one that will be quite straightforward to assess.
Fascinating. Okay, because there are certainly, many people with CFS are obviously searching for anything that might make a difference. So there's a lot of discussion about diet, and Isabel and Sue will say more about the Nutrition Club later on. But things like people have explored keto diets or FODMAP diets or all sorts of things. So I don't know whether there's anything you'd want to comment about any of those.
So the two biggest drivers of changing your gut microbes are an infection, GI infection. Actually, the three: medications, antibiotics, and also diet. So we know from human studies that if you change your diet, your gut microbe populations change you can detect change in within 36 hours. So they respond and react quickly to the change in your diet. And if that's a good thing, and you sustain that, then you'll be promoting that beneficial health benefit from that. But if you don't, so maybe this is an explanation for the yo-yo diet effect. You go on a diet to lose weight, but you don't stay on it. That when you come off the diet, the rebound effect is the microbes come back and think, God, we've been starved for so long. We now got to take advantage of going back to the original diet, and you gain the weight again. So your gut microbes respond very quickly to change in your diet, which can be a good or a bad thing.
So hence a bit of trial and error for some of that if the response is quick.
Yes.
Yeah.
Yeah.
Okay. In terms of the study, there's a set of questions about, well, there's several studies up there, but it's about whether people from across the UK can be involved, and I think the answer was yes, because a couple of them were online studies, and that sounded all possible. Are you selecting people, if 80% of people with ME/CFS have got some gut-related illnesses or difficulties, are you only selecting from that cohort? Do you use controls, and how do you get equal samples? So what's the criteria for getting them?
Okay, so we're currently just completing the inclusion-exclusion criteria for Restore Me, and we're aiming to have a homogeneous cohort of patients that have had the disease for similar number of years, similar severity, similar symptomology, and gut GI issues will be an important inclusion criteria. Doesn't necessarily have to be IBS, but some GI disturbance disorder, which is proven to be a chronic one, that would be an important inclusion criteria. Regarding stool collection, so we provide people with a stool collection device. We've been refining this over the years with various studies, and it is basically capturing your stool when you're on the toilet, putting it in an appropriate container, and then delivering that to the institute, or having it delivered to the institute. At the time when you deliver it to the institute, we'll then go through the other evaluation tests to assess the impact of the intervention. So it's a very straightforward process, and we provide the kit in order for you to collect and handle that material.
So the fecal transfer though is from healthy samples.
Ah, sorry. Okay. So yes, so we have an incredibly stringent criteria for donors.
Okay.
Not surprisingly, the donors are young, healthy people.
Mm-hmm.
Part of it is taking a sample from them and screening it for a whole variety of potential nasty things, but also doing psychometric evaluations as well. We want to know there's been no clinical issues like depression, illicit drug taking, those sort of things. We need lifestyle and behavior input because of the gut-brain axis. Okay, gut microbes can influence all these things or reinforce bad behaviors. We don't want to be transferring that into a patient. So very stringent criteria.
Fascinating. Okay, good. A couple of people, Leslie being the most recent, asking about any relevance to fibromyalgia. If you've got any kind of clues or indications.
Yeah, so again, there's a lot of overlap in the symptomology there. We are considering that as an inclusion criteria in the Restore Me because there could well be potential benefit there because of the link to the central nervous system and peripheral nervous system, as well as the nervous system in the gut, that can all be influenced by gut microbes. So yes, I think that could be potentially, FMT might actually have some beneficial impact on fibromyalgia as well, which would be an additional bonus, I think, for us.
That'd be fantastic for people to feel like they've got both some research happening and some potential clues. Right, I've got 4 bits here. Yeah, questions about the red lights.
Yes.
And, can people go out and buy red lights? Can they try it themselves?
No. So, well, I wouldn't recommend it because the lights have to have a specified wavelength.
Yeah.
Okay. Because the mitochondria in the cells in your body only respond to light of a particular wavelength. And those lamps are specified to deliver light of one wavelength. You go out and buy a red lamp, it's going to have a wide spectrum of red light. Only a very small component of which is of the beneficial wavelength. So no, going out and buying one on Amazon, etc., is not a good idea because there could also be harmful effects of getting exposed to other wavelengths in that red lamp.
Okay, so it's quite specific. So
yes.
Yeah, interest in timescales for a lot of these studies. Any ideas what some of the timescales are for what you have mentioned some of them in the talk? So
yeah,
it's any of the others really. And
so for the Light Me Up study, we'll be advertising for patient recruitment in December with the aim of starting the trial straight after Christmas, which will mean baseline assessments, sending out activity monitors, having patient go online and fill in the cognitive assessments online. They then mail back the wristwatches where we assess and bank the data. We then send out the red lamp. The red lamp will be used for 2 minutes every day for 2 weeks. At the same time, we'll be sending back the wrist accelerometers to assess any changes post-exposure to the lamp. they'll go online and complete the cognitive assessments again. So that whole study, although it seems, oh, it's like only 2 weeks, but actually collecting the data before and after and analyzing it, that could take a couple of 3 months. In the meantime, we'll be applying for all the regulatory and ethical approval for the Restore Me, with the aim of recruiting patients for that around between April and May, with a target start date of June, July. And again, that recruitment is through East Coast Community Healthcare. Patients registered with East Coast Community Healthcare will be eligible for consideration for inclusion. So you have to be registered with them.
Right. Okay. So that one's quite a local one.
Yes, East Anglia, Suffolk, Norfolk.
Yeah. Okie dokie. Right. Patricia's asked about red lights in saunas and spas, but I think you've covered that by saying it's a very, very specific wavelength that you're looking for. So not a general one. About you talked about depression being a kind of one of the exclusion symptoms.
That's the sort of endogenous depression, so for which serious medication has been taken. We don't include it entirely.
Okay.
But I'm leaving this to my clinical colleagues to decide what is and what isn't. Okay. So no, it won't exclude everybody that's ever had an episode of depression, because obviously that would mean we probably don't have many patients. But it's just, where do you draw the line? And that's down to my clinical colleagues.
Yeah, and that's obviously the concerns, because many people have been given diagnosis of depression, but that's not necessarily appropriate. But maybe it is. Who knows? Who knows indeed. Okay, I think we've covered pretty much many of the questions that have been asked by all our members. I'm just going to briefly hand over to Isabel and Sue from Cinnamon Days, if they'd like to ask anything further, but also perhaps to say a little bit about how the Nutrition Club works and how people can get involved. So I'm going to hand over to Isabel and Sue, but keep an eye on the questions.
Yeah. Can I just say there was a couple of questions on the Facebook feed. Can I just ask those first? These are both from Glenys. She said, I saw something with the red light therapy section that mentioned liver disease. Are you able to elaborate on that, please? And also, also, can autoimmune conditions set you up for ME, CFS?
Interesting questions. So the liver disease is one of the applications that red light has been used for, I think, principally in America. And that's on the list of FDA-approved use of the device. I'm not aware of anybody using it in the UK. I mean, in the UK, it's mainly used by athletes to accelerate muscle repair, recovery after excessive exertion, exercise, and also skin wounds. So some hospitals have used it to accelerate skin wound repair as well, but not really. So we'll be the first to try this in ME patients in a proper clinical trial setting. Your second question was about autoimmunity. So I know some of my colleagues consider ME CFS to be an autoimmune disease. Others will say, no, it isn't. Clearly, there are autoimmune aspects. If you look at symptomology and you analyze samples, you can identify what looks like autoimmune disease, so the production of antibodies that react with things they shouldn't be reacting in the central nervous system. But the key question there is that a cause or an effect of the disease? And we don't really know that. Interestingly, when we've looked at antibody responses to gut microbes as another potential cause of ME disease, there are clear differences in the antibodies from an ME patient and a normal healthy control, what they react to in the gut. And actually, in ME patients, there's less coverage. So there's immune deficiency, if you like. So ME patients can't react to the same profile of microbes that a healthy individual's antibodies can. So there's clearly some abnormality in antibodies in ME patients. The rituximab trial was entirely based around that, although a very extreme example of that. So yes, you could argue it's an autoimmune disease, it's triggering it, or autoimmune disease arises because of it. And I don't think we can really discriminate between those two at the moment. We just don't have enough data.
Okay, thank you.
Shall I just say a little bit about the Nutrition Club then, if that's all right? So Sue and I are co-directors of Cinnamon Days CIC, which is a social enterprise. And we have received funding from the Lottery Community Fund to set up a nutrition club specifically for people with ME, CFS or long COVID. And what we do is provide a free online nutrition club, which happens monthly, sometimes with events in between, like today's event. So we're trying to explore everything that we can around how nutrition can be of support and assistance to people who have these conditions. So it's free to join. Anybody can join. I've put the links in on the side in the chat. I'll put it on again in case anybody's missed that. But we're very, very glad to see any and all of you at future events. We also record our events. So if you're not able to make the event on the day, then you can join, you can catch up with it later on. So that's available for everybody as well. So I'll put the link up on the chat again just now, and you can please join us.
We've already had 9 new sign-ups while the talk's been in progress, so that's great. Thank you very much for joining.
Brilliant. Thank you very much, Sue and Isabel. I've got just a couple of last queries. One is, if people wanted to volunteer for any of the studies, Simon, how would they get how would they enroll? Is there a volunteer?
So if they go, if I could share my screen again. Let's share my screen and then go backwards. There we go. quadram.ac.uk/volunteer/.
Okay, so did everybody take a note of that? And I'll try and copy it as we speak into the chat. Okay, and there's a couple of questions flying around which are around, can vaccines disturb your microbiome and therefore have the potential to lead on to ongoing issues? People aren't specifically meaning coronavirus vaccines, they're meaning other vaccines as well.
So I think I've seen reports of ME patients not responding well to vaccines in terms of generating a full level of protection that non-ME patients would suffer from. So, and again, that relates to immune defects in ME patients, immune insufficiency or deficiency, if you like. So vaccines are usually injected. There is only one flu shot that's intranasal, I think. Oral vaccines, apart from the polio one. So because most vaccines are delivered by injection, they have less access to gut microbes, and therefore less chance or accessibility to the gut microbes influencing or changing the vaccine makeup. But I think ME can compromise the ability to respond to the vaccine. And there have been a couple of reports about certain types of vaccines maybe causing ME symptoms, but I don't think they've been substantiated. So that's probably not as clear. But certainly if you have a compromised immune system, irrespective of whether or not you have ME, you are not going to respond well to vaccines and get the full benefit of the vaccine.
Okay, thank you. So I've just put that link into the chat as well for anybody to copy, and perhaps we'll pop that link under our Facebook Live page. So I think we've had an amazing time with you. We've had a lot of your time. You've answered every question very, very diligently, for which I give you full marks. Thank you very much.
Thank you very much.
So a huge appreciation really to you, Professor Carding, for sharing your time with us and all the information and the slides. Would you be happy to share the slides with us so that people can access them?
Yeah. Would you like me to send a PDF version?
Perfect.
Otherwise, it's a huge PowerPoint, and I'm not sure people will be able to handle it, maybe not. So if I send it as a PDF version, that should be accessible to all.
Great, thank you.
Absolutely brilliant. And so a massive thank you to Cinnamon Days for jointly hosting this event with us. And we've had a fantastic number of people from a lot of places very widely across the globe, it seems. So obviously of huge interest to many, many people. So we're absolutely so immensely grateful that you undertake this research on behalf of a community that feels so research isolated. But it's just know there's an awful lot of people behind you and are really eager to hear
Thank you very
every
much.
bit of progress that you make. So a huge thank you to Professor Carding, and a thank you to absolutely everybody who's joined us this afternoon. You will be able to both watch this live on Facebook, and also you will be able to, when we've done it, which will be about 2 or 3 days, you'll be able, let's say Monday, we'll put it up on our YouTube channel so that you can watch both this one and also the one from 2.5 years ago, if you wish. So okay.
Very good.
Thank you.
Very good.
Thank you very much.
Thank you.
You're welcome. Thank you, everyone.